So thank you for the opportunity, and nice to meet you all virtually. So yeah, just got a small set of slides just to introduce you to the work that we do at the Medicines Discovery Catapult. We're based at the old AstraZeneca site, Alderley Park, just down the road from you all. And I wanted to give you a brief overview of some of the work we're doing. You'll see some parallels to what you've just heard from Azamina and Sam, but also hopefully some different aspects I can talk you through as well. Okay, so in terms of introducing the Medicines Discovery Catapult, we're actually the newest of the Catapult group. So we've been running for around 4 or 5 years, depending on when you call day one. The labs have been open for about 4 years now. And as Amina said, we're part of a network of catapults. We're a not-for-profit organisation funded by the government and our community. And because we got this financial independence, we can take risks and pursue high risk science, which is our key element of what we do, I believe. We work with a whole range of different types of collaborators. This is by no means an inclusive slide. We work with many SMEs and startups, but also work with academic groups. We work with large pharma on occasion, and we also work with technology providers, vendors, etc. So quite a diverse set of collaborators. But I think the one thing that brings this all together is a shared desire to improve medicines discovery by applying innovative science. Just got a couple of slides now from a larger deck, which is where we talk about who we help and how we can help different target audiences. And I've just pulled out a couple of slides which I think are particularly relevant for today. So in terms of how we can work with academics, sorry, I'll just move the picture slightly so I can read it. So we can provide impartial and unbiased medicines discovery project reviews, assessing the impact and potential of different innovations and products in the university. We can provide drug discovery insight and commercial know-how on the drug discovery process. We can provide insights into technology providers and identify partnering opportunities. We can deliver complementary lab work that we recognise as some really high-end technologies and capabilities in the academic sector, but on occasion we can deliver complementary science with technologies that aren't available within the university. We can apply informatics to optimise data that's generated and identify opportunities. And finally, as Sam outlined, we can help to identify grant and partnering opportunities. The other slide I thought is worth pulling out today is around how we can work with technology innovators. So these can be innovators that are developing new technologies within a university or in a technology-based startup. And what we feel with this group of collaborators is that we can provide drug discovery expertise to actually test those innovations with real-world biology and therapeutic examples. So that could be use of cells and tissues in a real-world context, or testing of compounds, just to bring that real-world testing to light. We can help to drive adoption of new approaches and technologies across the industry. So this could be working with you together on use cases and application notes. And often we can identify novel applications for technology. We can look across the sector and think about slightly different ways that a technology could potentially be used. But then we can benchmark your technology against other currently available gold standard technologies. In terms of how we help, just wanted to use this slide to introduce some of the different themes in terms of the way we work within Medicines Discovery Catapult. We have a large discovery organisation, which comprises Discovery Sciences, which is our lab groups. I'll go on to a little bit more on that in a minute. We have a theme around access to clinical samples. So we don't run our own biobank at the moment, but we've got relationships with different groups where we can help connect people to get the right samples for their research. And we have a virtual R&D organisation which offers project evaluation, project management, and we've also got a CRO network so we can help people to build a delivery plan for their project and help them to execute that delivery plan either through MDC labs or through connecting with CROs. We have a large informatics organisation which works on data science, artificial intelligence and machine learning, also bioinformatics and cheminformatics. And we have a syndicates theme, which is based around working with medical research charities and really trying to put the patient voice at the heart of the work that's being done so we can connect together different players around that medical research charity to try and drive an area forward. And I'll give you an example of that later. So going into the discovery department, which is where I work, I lead the in vitro lab activities within Medicines Discovery Catapult. I guess the key thing we're trying to achieve here is making a positive impact on the success of Innovators drug discovery in the UK. And the way we do this really is trying to generate decision-making scientific data and using a combination of high-end specialist equipment, specialist scientific know-how, but also drug discovery experience. We've got quite a large number of people who've worked in the pharma industry or in biotech, and we can bring that to bear on people's projects as well. In terms of what we do, we spent a lot of time really trying to talk to people and get under the skin of the project, understand what are the key problems, what are the key challenges, and trying to develop experimental solutions to help address those challenges. We develop a strategy, and then scientifically we've chosen to focus on 4 different areas. The first one is around preclinical cell models, trying to help identify, analyze, and advance better human preclinical cell models to test compounds and mechanisms in. Second one is around target and pathway engagement, helping collaborators to build that link between their lead molecule, the target protein, and downstream biology. Third one is around biomarkers, trying to help partners develop a biomarker strategy and apply our technologies to identify or validated biomarkers. And then the fourth one is around drug delivery and biodistribution through a combination of preclinical imaging, looking at whole body distribution studies, and then ex vivo tissue analysis to look in depth at where compounds are biodistributing in the body. To go into a little bit more detail, effectively what we're doing is operating between a simple 2-dimensional cell assay right through to analysis of clinical samples. And what we're looking to do is to help partners build the link between their compound, the target protein, and biological effect in a translatable system, ultimately to reduce project attrition. But we're also increasingly focused on back translation, taking clinical samples, really analysing these samples in depth and trying to translate that information back into the cascade. I've put a bunch of technologies that we focus on at the bottom, but in brief, what we've chosen to do is to focus in a number of areas. We've invested quite heavily in mass spectrometry, in spatial tissue analysis, in nanostring, and in microscopy, to name but 4. One thing I wanted to mention briefly is something we're very excited about, in that we recently were successful in gaining investment to build a new translational facility on site at Alderley Park. And this is going to be a lab or a set of labs where we can really drive research into infectious disease. So we're going to have level 3 biosafety facilities as well as level 2 labs, and we're going to be able to build access to strains and human tissue and cell models of relevance for people doing infectious disease research. And we've just brought in a head of infection biology. He's going to be joining us in a few weeks to drive this. So this will be a lab where we'll be open to either commercial or grant-funded work to help people drive forward their infectious disease research. I just wanted to finish off with 3 case studies just to paint a picture of the type of work that we've done. The first one is relevant to our discovery organisation, and this is work we've done with a spin-out at the University of Leeds called LUNAC Therapeutics. This was a group that was doing drug discovery initially from an academic setting at Leeds, looking to develop novel anticoagulants. And in 2019, we joined forces with this group for a biomedical catalyst grant. We were very successful with that grant, what a large award. And hopefully part of that success was the fact we could get our expert grant writer involved in helping to shape that grant. But then the element that we wrote into the grant for MDC was a little bit of a combination of industrial insert and project management and project planning, helping to connect into that CRO landscape I mentioned before, but also practically in the lab, preclinical imaging using novel PET tracers to look at mechanism in rodents. In 2020, I think in part due to this grant award, there was a large venture capital investment in the company. Data looked really promising, and then last year, LUNAC won Startup of the Year award at the BioNow Awards, and also received additional investment. So we're really proud of this type of example where we can help work with a company very early on and kind of help them on that early journey in whatever way we can. Second very different example I wanted to mention was from our informatics team. As I mentioned, we do a lot of work with data science and machine learning. And this is a piece of work we did with the MS Society. And what the MS Society wanted to do was to understand what the opportunity is for repurposing drugs, multiple sclerosis clinical trials. So what we did here is we started off with a list of compounds which had already been taken into trials for multiple sclerosis. We then used our algorithms and our databases to actually map back to the protein, the target proteins associated with these drugs. And then we look for other compounds which interacted with those target proteins. What we're able to do then is build a long list of drugs that could be considered as repositioning opportunities. And we pulled all this data together in an app or a desktop application that could easily be used to manipulate the data and look through the data. What was quite gratifying here is we were able to identify most of the candidates on the MS Society's initial list, but also identify a similar number of new candidate molecules which could be considered. And then the final example I wanted to mention was from our syndicates area, where we've built a psychiatry consortium to try and bring forward new targets for psychiatry drug discovery. So the way this syndicate works is it's again centered around medical research charities, but we've brought in a number of different pharma companies here, each of which have agreed to put funding into this consortium. And then what we do is calls for proposal from academics about new target opportunities, new target ideas in this space. It goes through a triage process, but then if these opportunities are taken forward, there's then the opportunity to fund research in the academic lab, in CROs, or in a combination of the two labs to build that validation package. And then there's an arrangement with the pharma companies where there's an opportunity to have a discussion at a later date about taking these things forward for commercialization. So that's just 3 examples I wanted to share quickly. So finally, just to wrap things up, hopefully I've illustrated there are a number of different ways that we can work with academics as they move their innovation forward. We like to engage very early through grant funding or other mechanisms, and then we can work with companies right through their journey, really, as they go through different funding rounds. We're very focused on trying to explore people's problems and find solutions, as opposed to have a tailored set of assays that we run routinely. And then, as I said, there's a number of different ways we can work. Very much, as Amina and Sam said, we can do collaborative work from our core resource, we can do grant-funded work, we can do commercial fee-for-service work, and we tend to just sit down with partners and really work through what's the best arrangement for the different experimental options. So I've put my colleague, Sander Hensing's contact details on the bottom. He leads our business development group, and he or I would be very happy to hear from you if you have any questions, either immediately now or after the meeting. Thank you very much.